Yes, next important essay question for carbohydrate metabolism is repeatedly asking question coming under essay question, coming under your practical charts, 10-mark charts, essay question, clinical scenario, everywhere it is coming. That is type 1 diabetes or type 2 diabetes, right? Sometimes nowadays gestational diabetes also mostly people are asking, right? For this type 1 diabetes and type 2 diabetes, you should thoroughly know about the regulation of blood glucose. Okay, for the regulation of blood glucose, the first point is we need a glucose supply for brain RBC and renal medulla. They expect only source is glucose for their energy and fuel. So maintenance and regulation of glucose is very, very important for this organ along with the others also. Right. We are having 2 points for the glucose. That is, one is entry point, another one is exit point. How the glucose is enters into the cell, how the glucose is coming out of the cell. Name the reactions like that, right? The first one is entry of glucose, you know very well, absorption of glucose. We are taking some polysaccharides, which will be converted into disaccharide, then finally monosaccharide. Monosaccharide is only able to uh, absorb the GIT. So, absorption from GIT. Second one, that is intestine. Second one is glycogenolysis. Already we are having some storage of glucose as glycogen in liver. So, whenever no glucose means this glycogen is able to converted into glucose, glycogenolysis. Third one is if there is no glucose supply in our body, we are ready to or we are able to synthesize glucose by the reaction of gluconeogenesis. Right? You know very well about the definition and the substrates for the gluconeogenesis, that is alanine, glucogenic amino acid, glycerol, lactate, pyruvate, like that. Okay. So these are all the 3 points for the entry of glucose. Likewise, the exit, that is factors removing glucose from blood. Number one is glycolysis. You all know very well, lysis means glucose should be lysed, glycolysis. Then they will go produce acetyl CoA, which enters into TCA cycle, then biological oxidation with the electron transport chain, then production of ATP. This is a routine one, right? Second one is glycogenesis. Excess glucose is stored in liver as glycogen. Third one is lipogenesis. Suppose beyond the limit we are having glucose, the excess glucose will go for the accumulation of lipids or production of lipids, lipogenesis. So these 2 points are your first descriptive factors. How to maintain the blood glucose? These are all the descriptive factors involved in regulation of blood glucose. Next one is you have to draw 2 diagrams, one for fasting state, another one is for fed state or postprandial state. That means 4 organs, they involved in the fasting state to maintain the blood glucose, they involved in the postprandial state to maintain the blood glucose. How? The first one is fasting state. The organ names 4 means liver, intestine, muscle, adipose tissue. Very, very 4 important organs: liver, intestine, muscle, adipose tissue. In fasting means there will be no food, so there is no place for intestine. You take out intestine, the remaining organs are liver, muscle, adipose tissue. How these 3 will produce some reactions and maintains the fasting blood glucose. First one is from the liver, glycogenolysis. Right? Liver releases glucose from glycogen. Second one is from the muscle, we are able to synthesize glucose by gluconeogenesis. Third one is adipose tissue. So in fasting situation, adipose tissue releases free fatty acids. They will go for oxidation of fatty acids and finally produces some amount of ATPs, that is energy. So these 3 are the fasting state factors, reactions, that is 4 organs produce 3 reactions, intestine rolled out. Whereas coming to the postprandial state, you are having some food. After that only these 4 organs will come for the maintenance of blood regulation. The first one is in intestine, the glucose is released, right? This glucose converted into glycogen. The reaction is glycogenesis in the liver. Second one is this glucose converted into lipogenesis and stored in triacylglycerol in adipose tissue. So, glycogenesis and lipogenesis. These two are coming under postprandial state. So, you have to draw two separate diagrams for one for fasting, one for postprandial state, right? In your book, it is clearly mentioned, very easy also, but descriptive point is very, very important. In postprandial state, there is no lipolysis and no gluconeogenesis. This is a very, very important point for your MCQ. Okay. Next, with this regulation, what is a normal fasting level? What is a normal postprandial level? You know, fasting means 8 to 10 hours fasting without food. Postprandial means 2 hours after food. So, in the fasting blood glucose, the normal level is 70 to 100 milligram per deciliter, whereas postprandial blood sugar level is 100 to 140 milligram per deciliter. You should mention or put one box. Within the box, you have to write these two normal levels, fasting and postprandial. Okay, these maintenance of blood glucose done by what are all the hormones? We know we are having two groups. One is hypoglycemic hormone insulin, another one is hyperglycemic hormone, that is glucagon, ACTH, GH growth hormone, thyroxine, epinephrine, like that. So you have to mention one or two reactions for this hyperglycemic hormones. Hypoglycemic hormone, the only function is it will decreases the glucose level insulin, right? After this, hormonal regulation and other factors regulation, the glucose is not properly regulated or controlled, means the disease is called as diabetes mellitus. Okay, so we have some one diagram or some descriptive points of homeostasis of blood sugar. That is how the organ respond to the two situations, that is like hypoglycemia and hyperglycemia. We have two situations, one is hypoglycemia, another one is hyperglycemia. Now, how the homeostasis of blood glucose will occur in our body? Suppose if you are having low blood glucose, what happens? It stimulates the pancreas, which releases the alpha cells, then they will release glucagon, right? This glucagon stimulates the liver, then the glycogenolysis, that means glycogen will be converted into glucose into the blood circulation, then normal blood glucose level will be attained or maintained. Okay, this is for low blood glucose. Suppose your blood glucose is high, hyperglycemia, what will happen? They again stimulate pancreas, stimulate the beta cells, releases the insulin. This insulin go to the peripheral tissues to stimulate the uptake of glucose from blood, and finally normal blood glucose level will be attained. This homeostasis of blood regulation is very, very, very important. You have to draw the diagram with the description for this homeostasis, then only the blood regulation essay question will be completed. Okay, bye.